everyone, my name is 𝗵𝗂𝗃. I am studying MZC second year and I am from Telangana Tribal Welfare Residential Degree College for Women, Janagam. So, my subject is microbiology, and in microbiology my topic is gram staining. So, the main objectives of my gram staining topic is introduction, history, types, importance, steps, requirements, procedure, result, and the last one is conclusion and as well as puzzle. So, let I discuss about firstly the introduction of gram staining. So, stains are natural or synthetic substances that can render the structure of cells visible. What is meant by stains, friends? Stains are natural or synthetic substances that can render the structure of cells visible, and gram staining is a method which is useful to differentiate bacterial species into two large groups. They are namely gram positive and as well as gram negative. So, each and every cell is made up of a protective layer called cell wall. So, based upon cell wall composition, the bacterial species are divided into two types. They are namely gram positive and as well as gram negative. So, in gram positive bacteria, 90% of the bacterial cell walls are made up of peptidoglycan layers. So, when we compare it to the gram negative bacteria, only 10% of the bacterial cell walls are made up of peptidoglycans. So, what is peptidoglycan, friends? Okay, the peptidoglycan is a layer which can considered as backbone of a bacterial cell wall. So, in this peptidoglycan, it consisting of mainly two portions. They are, the first one is glycan portion and the second one is peptide portion. So, in glycan portion we contain two alternating units. They are namely NAG and NAM. So, what is meant by NAG, friends? NAG means N-acetyl glucosamine and NAM means N-acetylmuramic acid. So, these are the two alternating units which are interconnected by beta-1,4 glycosidic linkages. So, due to the presence of these peptidoglycan more in gram positive bacteria, so this is the peptidoglycan layer we can find in gram positive bacteria. Due to the presence of these, the bacteria can absorb the stains more when we compare it to the gram negative bacteria. Actually, what is meant by stains? Exactly, the stains are colored substances which is useful to visualize the microorganisms under microscope. So, the organisms which can't be seen with our naked eye, it can absorb only under microscope. Those organisms are known as microorganisms. In our surroundings, we can find different types of microorganisms. In that, 90% of the microorganisms are useful to us, whereas only less amount, it means 10% of the microorganisms are very harmful to us. They can cause infectious diseases such as cholera, dengue and etc. So, this is the introduction about these gram staining. In this picture, we can find differences between gram positive and gram negative cell walls. Here, it represents the peptidoglycan layer. The peptidoglycan layer is more in gram positive bacteria. For this reason, the gram positive bacteria absorb the stains more when compared to the gram negative bacteria. So, in this, it is a layer, it is known as cytoplasmic membrane, and the space between these peptidoglycan and cytoplasmic membrane is known as periplasmic space. So, this is the periplasmic space. And the next one is history. History of gram staining. A Danish scientist called Hans Christian Gram. A Danish scientist called Hans Christian Gram developed this technique while he is working along with 𝗂𝗽𝗏𝗏 in the morgue of the city hospital in Berlin in the year of 1884. So, this is the history of gram staining. Next, bacterial stainings are different in types. They are majorly, the bacterial stainings are divided into two major groups. They are, the first one is simple staining and the second one is differential staining. So, in simple staining, we can visualize the morphological shape and arrangement of bacteria. In simple staining, what we can visualize in simple staining, friends? Yes, exactly. We can visualize the morphological shape and arrangement of bacteria in simple staining. So, the second one is differential staining. In differential staining, it is again broadly classified into two types. They are identification and visualization of structures. So, gram staining and as well as acid-fast staining, these are the two stainings comes under identification in differential staining. So, whereas visualization of structure of bacteria, we can find four different types. They are namely, the first one is flagellar staining and the second one is capsular staining and the third one is wall staining and the last one is spore staining. So, these are the four different types of stainings we have under visualization of structure of bacteria. So, if we want to observe the flagella in a bacterial cell, we can find flagellar staining is necessary. So, what is flagella, friends? Obviously, flagella is a locomotory organ which is raised from the cytoplasm of a bacterial cell, and these flagella plays a major role in locomotion. So, what is meant by locomotion, friends? Exactly, locomotion means the movement of organism from one place to another place is possible with the help of these flagella. So, in this cell wall we can find this is the flagella. So, this structure is known as flagella, and the next one is capsule staining. If we want to observe the capsule, then the capsule staining is necessary. So, capsule means bacterial cell walls can shed their polysaccharide layers. These polysaccharide layers consisting of high molecular weight. What, friends? These polysaccharides layers consisting of high molecular weight. So, due to the presence of these high molecular weight, the bacteria can form a layer around a bacterial cell wall. So, this is the layer which we can find, it is known as capsule. If this layer is tightly packed with each other, it is known as capsule. In this picture, we can find the capsule. Yes, this is the capsule layer or in a bacterial cell. If these polysaccharide layers are loosely packed with each other to form a layer is known as slime layer. So, if we want to absorb the capsule in a bacterial cell, capsule staining is necessary. And the next one is endospore staining. Endospore, these plays a major role in protection of bacteria. These endospore can protect the bacteria under environmental conditions also. So, these are the different types of organelles present in bacteria. They play a major role for the protection of bacteria, like flagellum. So, this is the flagellum, and these bodies are called inclusion bodies, and these rod-like structures are known as plasma, and these finger-like projections are called pili, and this layer is known as capsule. And the next one is importance of stainings. So, the microscopical examination of a stained smear can enable us to learn about the morphology, relative sizing and arrangement of bacteria. So, the bacteria can exist variety in shapes. For example, these are the different types of bacteria which we have. The bacteria is mainly divided into 4 different types. They are namely 𝗂𝗽𝗱, 𝗬𝗫𝗧, 𝗬𝗦𝗔𝗧 and as well as 𝗵𝑅𝑖O. The bacteria which exist their shape in the form of round like structures is known as 𝗂𝗽, and the bacteria which exist in the form of long rod like structures is known as bacilli. If the bacteria are spiral in shape, it is known as spirillum, and the bacteria which exist in the form of comma shape, it is known as 𝗵𝑅𝑖O. So, what are the different types of bacteria we have, friends? There are 4 different types of bacteria. They are 𝗂𝗽𝗱, bacilli, spirillum and as well as 𝗵𝑅𝑖O. So, under 𝗂𝗽, based on arrangement, in 𝗂𝗽 there are again divided into 4 different types. They are namely, the first one is monococcus and the second one is diplococcus, the third one is streptococcus and the last one is staphylococcus. So, these are the different types of bacteria in 𝗂𝗽. So, if these 𝗂𝗽 are arranged as a single in layer is known as monococcus. If 2 𝗂𝗽 are attached with each other to form diplococcus, and the third one is streptococcus. More than 2 𝗂𝗽 are attached with each other to form a layer like structures. This is known as streptococcus. Or more than 2 bacteria are attached with each other to form a chain or branch like structures is known as streptococcus. And the last one is staphylococcus. It means more than 2 𝗂𝗽 are attached to form a clusters of cells like structures. These are known as staphylococcus. So, these are the different types of bacteria based on arrangement. It can be divided into 4 different types in 𝗂𝗽. So, in bacilli, it is also present in different types. For example, 𝗂𝗽 bacilli. And in spirillum also, the bacteria which exist in spiral in shape, it is known as spirillum. So, then it is the importance of staining. It is also helpful for the detection of cells. For example, especially the pus cells and polymorphonuclear cells. What, friends? It is these gram staining is also helpful in the detection of cells such as pus cells and especially polymorphonuclear cells. And bacteria can also be differentiated by using these staining reactions. For example, gram positive from gram negative organisms. So, this is about the importance of staining. I recap it once again. Importance of staining. So, the microscopical examination of a stained smear can enable us to learn about the morphology, relative sizing and arrangement of organisms, and these plays a major role in the detection of cells such as pus cells and polymorphonuclear cells. As well as these gram staining is also useful to differentiate the bacteria into gram positive from gram negative organisms. And the next one is there are different types of steps involved in gram staining. There are majorly 4 different types of steps involved in this gram staining. The first one is primary staining. It is nothing but crystal violet. And the second one is addition of mordant. It is nothing but grams iodine. And the third one is decolorizing agent. Decolorizing agent which we have used in this gram staining is alcohol or acetone. And the last one is addition of counter stain. It is safranin. So, these are the 4 different types of steps involved in gram staining. Let I recap it once again. There are 4 different types of steps involved in this gram staining technique. They are primary staining, it is crystal violet, and the second one is addition of mordant, it is grams iodine, and the third one is decolorizing agent, it is alcohol or acetone, and the last one is addition of counter stain. The counter stain which is used in this gram staining method is safranin. And next, the requirements used in this gram staining are a heat fixed bacteria, and the next one is crystal violet. Yes friends, it is crystal violet. And the second one is grams iodine. So, this is the grams iodine which is used in gram staining technique. And the third one is acetone. So, this is the acetone which I have used in this gram staining technique. And the last one is safranin. It is termed as counter stain. This safranin is used in gram staining technique. So, these are the 4 types of requirements which we have used in this gram staining technique. So, along these crystal violet, grams iodine, acetone and safranin, other requirements are also used. They are commonly called as inoculation loop. So, this is the inoculation loop which we have used for culture, and there are different types of glass slides which we have used in this gram staining technique. So, these are the different types of requirements used in gram staining technique. And the next one is procedure. So, prepare a heat fixed smear of bacterial culture. Okay friends, here what is meant by culture? Yes, culture means cultivating of microorganisms. So, cultivating of microorganisms is termed as culture. Preparation of culture is under only flame, because this flame can prevent the contamination of other microorganisms when we perform this procedure. So, with the help of inoculation loop, so this is the inoculation loop. With the help of this inoculation loop, take 0.1 micro ml of culture, and this culture is placed on a glass slide. So, the spreading of culture is known as smearing. What friends? The spreading of culture is known as smearing. After this smearing, this smear with crystal violet and wait for a minute. Then the next step is add grams iodine which washes the crystal violet stain. After that, rinse the slide in running water and add decolorizer. The decolorizing agent which we have used in this technique is alcohol or acetone. So, after rinse the slide in running water, add decolorizing agent. It is nothing but alcohol or acetone. And again rinse the slide and cover the smear with safranin, and after that wait for a minute. After that, wash off the safranin with the water and air dry the slide. So, when we observe this slide under oil immersion lens, this is the procedure of gram staining. So, this is the practical representation of procedure in gram staining. In the first step, we can add crystal violet. So, after the addition of crystal violet, wait for a minute and wash the slide. After that, add grams iodine, wait for a minute and wash the slide. The third step is addition of decolorizing agent. So, the decolorizing agent is alcohol and acetone. 95% of ethyl alcohol is added and wait for 5 to 10 seconds. After this time, wash the slide and add safranin to that slide. After 1 minute and wash this slide and observe under microscope. So, when we observe under microscope, we can find different types of microorganisms. For example, in this picture, we can visualize many different types of cells. Some of the cells are long rod-like structures and the other cells are round in shape. So, if the long rod-like structures are termed as bacilli and the short structures which are in shape is termed as bacilli. Why? We can observe two different types of colors here. They are violet and purple. The bacteria which absorbs violet color is known as gram-positive bacteria and the bacteria which absorb pink color is known as gram-negative bacteria. So, here we can find two different types of bacteria. They are gram-positive and as well as gram-negative. So, why these gram-positive bacteria absorb purple color? Here means it is consisting of peptidoglycan layer. So, in gram-positive bacteria, these peptidoglycan can act as a permeability barrier. It means barrier means it can stop the things. So, in gram-positive bacteria, this peptidoglycan is act as a permeability barrier. So, this barrier can prevent the loss of crystal violet iodine complex. So, by this stopping of crystal violet iodine complex, when we add alcohol to these gram-positive bacteria, this alcohol, the main function of this alcohol is to dehydrate the thick layer of peptidoglycan. So, when the peptidoglycan layer is dehydrated, it leads to shrinkage of pores. So, shrinkage of pores appears on the surface of peptidoglycan layer. By this shrinkage of pores, the bacteria remain purple in color. So, this is the reason behind that why gram-positive bacteria are purple in color. So, this is the reason why bacteria are purple in color. And the next one is gram-negative bacteria are pink in color. So, why gram-negative bacteria are pink in color? Let me discuss. So, means the gram-negative bacteria, when we add alcohol to these gram-negative bacteria, these can remove the crystal violet iodine complexes. So, when these removes the crystal violet iodine complexes, these gram-negative bacteria remain purple in color. So, these are the different types of bacteria which we have, and in gram-positive and negative bacteria are also, they are again divided into major types. They are gram-positive 𝗂𝗽, gram-positive bacilli. So, the main examples of gram-positive 𝗂𝗽 and gram-positive bacilli are the main gram-positive 𝗂𝗽. It means streptococci, staphylococci, actinomycetes, lactobacillus. These are the different types of gram-positive 𝗂𝗽 and gram-negative 𝗂𝗽. It means Neisseria is the best example for gram-negative 𝗂𝗽. So, these are the different types of bacteria which we have comes under gram-positive and gram-negative 𝗂𝗽. And the last one is, friends, here it is a puzzle for you. Think for a while and try to solve this puzzle. Is there getting any ideas about this puzzle, friends? Absolutely. Here come in and stain. In this puzzle, C stands for crystal violet and I stands for iodine, A stands for acetone and the last S stands for safranin. So, these are the requirements used in this gram staining technique. This puzzle may help you for easily remembering of the requirements which are used in this gram staining technique. So, this is about my topic gram staining. Let me recap what we have learnt. Stains Stains are natural or synthetic substances that can render the structure of cells visible, and gram staining is a method which is useful to differentiate the bacterial species into two large groups. They are commonly called as gram positive and as well as gram negative. And the next one is, we can use 4 different types of steps involved in this gram staining technique. They are majorly, the first one is primary stain, it is crystal violet, and the second one is addition of mordant, and third one is decolorizing agent, fourth one is safranin. So these are the 4 different types of steps which we used in this gram staining technique. So I specially thank to TSAT for this program, and I also thank my mentor teacher Mrudula madam and as well as Saujanya madam. I especially thank to my principal madam Dr. Haripriya madam, and I thank everyone, each and everyone who supported me. Thank you.